Dear students, today we are going to discuss about the topic immunoelectron microscopy and surface plasmon-resonance. Both the topics belongs to the module 3 of the course MBT. First, I will start with immunoelectron microscopy. Immunoelectron microscopy or IEM can be defined as technique that uses antibody that interact with antibodies, for example, protein A or protein G, in conjunction with electron microscopy to localize ultrastructurally antigen or the antibodies that is present in cells or in the tissue section. So it is one of the best method for detecting and also localizing proteins in the form of antigen or antibody that is present or expressed in cells or tissue section. In addition, a number of other biological macromolecules whose specific ligand binding properties are known can be used in this process, such as lectins can also be used for immunoelectron microscopy technique. Immunoelectron microscopy was a term that was originally confined to studies that made use of the transmission electron microscope, but now include those that use the scanning electron microscope also. So this particular procedure can be used on particularly practically every unicellular and multicellular organism, and often provides unexpected insights into the structure function association. This particular technique uses transmission electron microscope for the visualization purpose, and in the current days, scanning electron microscopes are also in use. So ultra thin section of the tissues are labeled with antibodies against the required antigen and then labeled with gold particles in the form of a secondary antibody conjugate. So gold particles of different diameters enable two or more proteins to be studied simultaneously. So in transmission electron microscope, electrons serve as a pseudo light source. High energy electrons emitted from the filament that are accelerated and forced by electromagnets into a very fine coherent beam in the center of the microscope column. And because electrons are used, the microscope column and specimen area are maintained in a vacuum condition to prevent the deflection of electron by atmospheric gases. So the electron beam is focused on to the sample, and here the more electrons preventing their passage through the sample, those electrons with enough energy to penetrate are transmitted, further focused and projected onto a phosphor screen. So the image is resolved on the phosphor screen due to the emission of photons in the area that's stuck by the transmitted electrons. The image can be permanently recorded directly on photographic film sensitive to electrons or visualized with photographic cameras interfaced with the electron microscope viewing chamber and a computer to rapidly generate high resolution computer processed image. So this particular technique is a method of performing an immunohistochemical method using an optical microscope in an electron microscope known as various names such as immunocytochemistry and electron microscopic immunohistochemistry. Like immunohistochemistry is a different thing where we are screening the component the tissue by using antigen antibody reactions. Here the antibodies are used in that are fluorescent level cases. But what we are using here in immune electron microscopy, the principle is different. Here, instead of fluorescent probe with the secondary antibody, what we are conjugating in immunohistochemistry process, but here in immune electron microscopy, we are using immunoglobulin conjugated secondary antibody, and this particular process is known as immunoglobulin leveling. So here the principle is that just like the simple electron microscopy, we have to take carbon coated copper grid, and over the grid, first we will use the sample solution, means the antigenic solution, purified antigenic solution. And over the purified antigenic solution, we have to use its specific primary antibody. Means primary antibody that is specific towards this antigen. So here antigen antibody complexation will happen. And before that, when we are coating the carbon coated copper kit with the antigen solution, after that we must use some blocking agent as we are using in immunofluorescent technique only to restrict untreated attachment of primary antibody. So first we are using the blocking solution, then we are adding the primary antibody. And with this, then we have to remove the unbound primary antibody, wash it, and then we have to add a gold conjugated secondary antibody that is specific to the primary antibody. Here, colloidal gold particles are most often used that are attached to the secondary antibodies, which are in turn attached to primary antibody designed to bind a specific antigen or other cell component. So here the solution may be a cell that has the antigenic surface or a simple specific antigen isolated from any species. Gold is used here for its high electron density, which increases electron scatter to give high contrast dark spot. So in immunostaining, in immunohistochemistry or immunofluorescence staining technique of tissue section, so that the cells can be visualized under the microscope, there we are using, in case of the optical microscope, means fluorescent microscope or confocal microscope, we are using fluorescent probe. Here we are not using any fluorescent probe, instead we are using gold because it will increase electron scattering and henceforth it will develop high contrast dark spots. So the structure and the location of the antigen that can easily be detected. So another method is used here, which is known as immunofixation. So immunofixation is the method that is used for the identification of antibodies for a specific antigen. So that method is known as immunofixation process. So here, the success of immunogold leveling technique. So how the immunogold leveling technique is helping in detecting the antibodies via immunoelectron microscopy technique. It depends on these three parameters. The success of immunogold leveling technique depends on quality of protein antigen preservation, antigen and primary antibody specificity, and the antibody's ability to infiltrate cells and the tissues. So these are the three criteria that are prerequisite for the success of immunogold leveling technique. Now, how this immunogold leveling technique occurs? It's a procedure of IEM. So in preparation for transmission electron microscopy, samples are usually fixed with aldehydes. So what type of aldehydes we can use here? We can use formaldehyde, butyraldehyde, or a combination of the two. And after the aldehyde fixation, means post-aldehyde fixed in osmotic in osmic acid, N block stained with uranyl acetate solution, dehydrated with alcohols, means different percentage of alcohols, and embedded in a resin. The embedded samples are thin section with a glass or diamond knife and subsequently counterstained with heavy metal ions like uranium and lead salt. So here uranium ion means uranyl acetate and lead acetate solutions are used to further enhance the electron density of positively or negatively charged molecules in the sample. So the specimen thus prepared have very good contrast and can be used to observe and record the molecular and subcellular details of cells and tissue section. Alternatively, molecules can be absorbed in a thin plastic film, followed by embedding in an electron-dense stain or angular shadowing with evaporated metals. So, immune electron microscopy is used to localize molecules at the ultrastructural level by leveling them with specific antibodies. So here antibody leveling requires some steps. So before adding the antibody, we have to block the antigen solution. Antigen coated copper grit. Because those portions where the antigens were not bound with the grit, those portions should be blocked. So here, just like immunofluorescence process, we have used BSA as a blocking agent. Here we can use as a blocking agent. After that, we have to add primary antibody. And this primary antibody should have specificity towards the antigen. Next step is adding secondary antibody with gold conjugate. So here secondary antibody means the labeling can be done by pre-embedding or post-embedding process. So what is pre-embedding and post-embedding process? I'm coming to. So here you can see that this process is the pre-embedding process. Here fixation, first we are going for fixation using formaldehyde and butylaldehyde, or maybe one aldehyde. Then we are going for permeabilization by using Triton X100. It's a detergent, and then we are blocking the sample using serum albumin, BSA. After that, we are going for immunogold leveling, adding primary antibody and then colloidal gold. So embedding, next step is embedding using some epoxy resin. After embedding, we have to go for sectioning, and only the sections having the thickness of 50 to 70 nanometer can be used for further processing or further negative staining. Here, negative staining can be performed using uranyl acetate solution as well as lead acetate solution in a dehydrated chamber. Then we can dehydrate it and observe it under the transmission electron microscope. In case of post-embedded embedded technique or post-embedding process, after fixation, we are going for embedding. And here we are not adding epoxy resin. Instead, we are using acryl resin. Then we are going for sectioning. Similar thickness tissue sections would be preferred. And after that, we are getting the tissue sections, very thin tissue section, ultra thin tissue section. We are using blocking agent, means PSA. Then we are going for immunogold leveling and observation under the transmission electron microscope. So this is the difference between pre-embedding and post-embedding. What can be possible? So this different technique means pre-embedding and post-embedding immunogold leveling. In case of pre-immunogold leveling, which is primarily used for the detection of proteins, antigens, and other macromolecules of our interest that are located on the surface or the exterior of cells. Virus particles and other extracellular biological specimens can also be observed using pre-embedding process. In case of post-embedding leveling technique that are used exclusively for the detection of proteins, antigens, and other macromolecules of interest that are located at the interior, not at the surface or exterior of the cell, that are located at the interior. There we can use post-embedding leveling, that means immunogold leveling process. So at the interior or intracellular region of the cells, virus particles, and other extracellular biological specimens, we can use the post-embedding immunogold leveling process. So that is all about the process of immunoelectron microscopy. Now I'm coming to the application part. Immunoelectron microscopy was developed as a diagnostic aid for the detection of and detection and identification of transmissible gastroenteritis virus, rotavirus that is present in fecal and intestinal content, from cases of gastroenteritis in young pigs. Again, IEM is one of the fastest and most sensitive method for the detection of and diagnosis of viruses like the plant virus, animal viruses such as hepatitis virus. So hepatitis A virus. So this particular technique is very specific, sensitive, and also quantitatively we can assay a hepatitis A antigen and antibody of hepatitis A. Anti-HA. It's practically for large-scale testing is limited. So mostly for anti-HA or hepatitis A antibody determination and antigen determination. This method is very specific, sensitive method, and quantitatively we can analyze it. So that's all about immunoelectron microscopy. Next, I'm going to the second technique, that is surface plasmon resonance. Plasmon is a word that means that the collective oscillation of the free electron gas density open at optical frequencies. So surface plasmon resonance means that is one of the commonly used technique for detailed and quantitative studies of protein-protein interactions and determination of their equilibrium and kinetic parameters. So surface plasmon resonance provides excellent instrumentation for a level-free, real-time investigation of protein-protein interactions. So if you look into the principle of SPR, so this particular technique is actually based means SPR-based binding method involves immobilization of the ligand on the surface of a sensor chip, which has a monolayer of carboxymethylated dextran that is covalently attached to a gold surface. The ligand of interest is immobilized on the surface of the sensor chip using well-defined chemistry, allowing solutions with different concentrations of an analyte to flow over it and to characterize its interaction to immobilized ligand. So here you can see this is the gold surface, and over the gold surface a monolayer of carboxymethylated dextran is present, and this as a whole function as a sensor chip, and over that the ligands are attached, and this ligand means which is present in the analyte. So there may be interaction between the ligand and the analyte, and here this is in this direction this is the flow of analyte, and it will be characterized by the interaction between the ligand and the analyte. Means the ligand that is immobilized over the sensor chip surface, and it will interact with the analyte. So how it is interacting, we can detect the interaction between the two ligand and analyte using this particular instrument or using this particular experiment. So the SPR signal, which is a typical signal that we can get from SPR measurement, mostly for protein-protein interaction, antigen-antibody interaction. So this particular signal originates from changes in the refractive index at the surface of the gold sensor chip. The increase in mass associated with the binding event causes a proportional increase in the refractive index, which is observed as a change in response. These changes are measured as changes in the resonance angle. So the resonance angle here the resonance angle is delta and theta. So this resonance angle of the refracted light, when the analyte, which is flowing in a microfluidic channel, binds to the immobilized ligand and increases in density at the sensor chip surface. Next, importantly for protein-protein interactions, this may be cell-cell interaction or simple antigen-antibody interaction or simple protein-protein interactions. The change in refractive index on the surface is linearly related to the number of molecules bound. So the response signal is quantified in resonance unit, and represents a shift in the response angle, where one response unit is equal to a critical angle shift of 10 to the power 4 degree, or 10 to the power minus 4 degree, or 10 to the power minus 12 gram per millimeter square. So when a steady state is achieved, all binding sites, means steady state means all binding sites are occupied. The maximum resonance unit is determined uh, using a formula, which is Ru is equal to NRU uh, into MWA by MWA. So here, monitoring the change in SPR signal over time produces this type of sensorgram. Means it's a plot of the binding response, that is the resonance unit in the term of resonance unit versus uh, time, which allows different stages of a binding event to be visualized and evaluated uh, in this figure. So during the injection of an analyte, the binding response increase is due to the formation of analyte ligand complexes at the surface of the sensor chip, and the sensorgram is dominated by the associated phase. After a certain time of injection, a steady state is reached. Here you can see. So this is association between two type of. So this is one protein, and this Z1 is another antiprotein, maybe antigen antibody, or maybe antibody with secondary antibody. So it's a protein-protein interaction. So in this step, you can see it's constantly increasing. Means this is the association step. So here we are getting the resonance signal, and we can calculate it in the form of resonance unit. Now, so the kinetics is going on, and after that you will get some plateau-like structure. So when the molecules are in association, so after a certain time interval, time point of means after a certain time of injection, a steady state is achieved, where binding and dissociation molecules are binding and dissociation both are in equilibrium. So the decrease in response after analyte injection is terminated is due to dissociation of the complex. So defining the dissociation phase. So this phase here, you can see that this is constantly there is a fall. So this fall is actually the dissociation between the two proteins, and this phase is known as dissociation phase. Depending on the dissociation rate of the tested ligand, some assays may require a regeneration step in order to reach the baseline again. So this process is known as the regeneration. This step is known as regeneration, or this phase is known as the regeneration phase, regeneration step. So fitting the sensorgram data to an appropriate kinetic binding model allows calculation of kinetic parameters such as association constant and dissociation constant, rate constant, and also binding affinity that should be tested during this interaction study. So here we have observed there are three parameters from the sensorgram data. One is your kinetic one, and the light concentration and diffusion rate. So the kinetic could also be analyzed from the sensorgram for different analytes which have same affinity with the same ligand would have different kinetics. So the analyte concentration should be confirmed before final SPR experiment, which is critical for the experiment. And the concentration range should span uh, 10 times below the dissociation constant and 10 times above the dissociation constant. The diffusion rate could also affect sensorgram result. If the diffusion rate is lower than the association rate, then mass transfer will affect effect can be observed in the typical signal what we are getting from the SPR. So to operate this experiment or the procedure for the experiment, there are 6 to 7 steps. The first step we will in the first step we will see that first you have to express and purify the ligand and the analyte. This is the first step. Next step is choice of appropriate sensor. Then determining the pH and ligand concentration for optimal immobilization. So dilute protein sample in buffer prepared at several pH following the surface preparation process and choosing the condition of a higher surface concentration of ligand on the chip. So this is a trial and error method. You have to determine the pH ligand concentration that is optimum for immobilization. Next step is regeneration scouting and surface performance test. So following regeneration scouting and surface performance test, then you have to select regeneration method to run the experiment. After that, we have to select binding analysis and direct binding to investigate protein. So protein binding means kinetic analysis and mass transfer to run kinetic assay accompanied with binding experiment should be assessed. And all of these data should be analyzed and kinetic constant should be determined. So this is all about surface plasmon resonance. Now, where we can apply this particular process or particular experiment? So mostly we use for thermodynamic analysis or protein-protein interaction. SPR can be used in different biological. It has different biological applications, such as in biosensors for specific biological interactions, including absorption and desorption kinetic study, antigen antibody binding study, epitope mapping for determination of biomolecular structures, antigen antibody structure, and interactions between the proteins, DNA, protein, and then viruses, and also in the study of lipid bilayer, we can also use surface plasmon resonance. For non-specific biomolecular interactions, means for the study of biocompatibility of any particular compound, we can use surface plasmon resonance. That's all.